Science Research Experience 2025. I know most of you, but not everyone. My name is Eleanor Harris. I'm one of the co-directors here at Clifton. I've been working with the students this week, along with Bridget Bradshaw and Cheyenne Hawkins, who are our educators. Thanks very much for coming. I say this every year, but it's true every year. This is my favorite week of the year. It's astonishing every year to get to work with such curious, open-minded, hardworking students and see them follow their curiosity and work really hard and then pull together really interesting results. It's really a joy every year. I just wanted to kind of give you an overview of what we've done this week to show you how much they get done in a very short amount of time. So most of them come Monday morning with maybe there's some ideas about what you want to do, but pretty open to the wonders of the world. And so on Monday morning, we go out for a hike, see what kind of cool bugs or plants or fungi we can find. And then by the end of the morning, they usually have really deep, insightful, interesting questions. And then the entire time for collecting data is from Tuesday morning until yesterday around noon. And that was even curtailed a little bit this year because we were trying to keep people inside out of the sun. So a very short amount of time to collect data, which they will tell you all about how they did. And then they have been working on these presentations since about noon yesterday. So they pulled together pretty incredible stuff in a very short amount of time. So I hope you'll be as impressed with them as I am. Am I forgetting anything? No. Okay. We'll get right to it. So our first young scientist is Lorelei Nason, who will be telling us why autumn olive leaves have glitter.
Hello, everybody. My name's Lorelei. I am a rising 11th grader, and I go to Germanna Community College. I am a competitive rock climber, and this is my 4th year doing this camp. So I studied autumn olives, which they are an invasive bush from Asia that came here in the 1830s. I've been doing this for forever, and I did the what was the thing called that I did over the summer? Yeah, I did Nature Club, and we had one day that we did nature journaling, and I decided to do autumn olives, which is this photo here. And they have this glitter on their leaves that I was very interested in about why they had this glitter. So this is also another photo of the glitter under a microscope that I took. And when Googling it and researching, I couldn't really figure out why they had this glitter. And they're also known as silverberry because of the scales on the berries, like you can sort of see in the photo. So my research question was, why do autumn olive leaves have glitter? And now I have a lot of hypotheses about this. So my first one was that it might be insect protection to help protect them from getting eaten. The second was that it might be sunscreen protection so they don't get sunburnt, because leaves can get sunburnt. The third was that it would help protect from losing moisture, which is pretty important for plants. My fourth one is that that I thought that the newer leaves would have more glitter because they haven't been as exposed to elements. So like when all the storms we got and stuff, that would knock some of the glitter off. So I thought the newer leaves would have more. My fifth one was that I thought the plants could be affected by how much was nitrogen and some other things were in the soil, because autumn olives, because they are invasive, they put nitrogen in the soil and they screw up the soil levels. So I thought maybe that had something to do with it, but I didn't really know. Because of the timing restraints, I was only able to do the first 4, and I couldn't study the wind or the water, so I just did those 4. So to test everything, for the insect protection, I measured how much of a leaf was eaten and the amount of glitter that was on that leaf. For the sunscreen, I measured where the leaf was on the plant, where the plant was in the property, and the amount of glitter on the leaf. For moisture, I measured the amount of glitter on the tops of the leaves and the bottoms of the leaves. And for age, I measured the amount of glitter on young and old leaves. So the glitteriness was determined by a scale from 1 to 6. It was originally 1 to 5, but of course we got outliers and I had to change it. Due to the wind, it made it pretty hard to get results. So what I would take a leaf and scratch it on the paper like this and see how much glitter came off. But it was pretty windy the first day, so like a lot of it was getting blown off. I was like, I don't know what's going on here. And it was pretty dewy in some of the mornings, so I'd also go and I'd just get a wet smudge and be like, well, that was not helpful. I also measured the age. I chose a branch in the plant and numbered the leaves starting at 1 and moving outwards. It'd be like 1, 2, 3, 4, 5, all the way up to wherever the last leaf was. So newer leaves have bigger numbers and older leaves have smaller numbers. So this is my study sites. These black circles is where I was. So this up here is Upper and Lower Woodcock. This is all Turkey Gap, and the brown line is the road. So I also was up on the road a little bit. Because it's invasive, it's sort of everywhere. So like I found it literally everywhere. There's some bushes like right down there. There's some right across the pond. It's just sort of everywhere. I tried my best to get samples from everywhere, but it was so hot. We tried to stay in the shade, so it was a little biased there. So here's graphs. So my first was how much does the bugs affect? Does the glitter affect how much bug is eaten? So this over here is the percentage of the leaf eaten, and this is the level of glitter on the bottom from 1 to 6. Your eyes like to trick you and say there might be a line going like this, but this is really just saying there was no evidence supporting any of this. This graph is showing where the bushes were. So if they were in the open fields with a lot of sun, if they were on the edges of trails or edges of woods with a decent amount of sun, and this was out in the middle of the forests. So again, this is glitter level on the bottoms of the leaves and where they were. So this shows that I thought there would be a lot of glitter out in the fields because maybe it would be like sun protection, but this is showing actually there was more glitter when it was out in the middle of the forests. This graph is a little chaotic, but this is showing the glitter level on the bottoms of the leaves again, and the position on the bush. So again, the lower numbers, so like the 2s and 0s down here, this is the older leaves. This down here is the younger leaves. So you can see the older leaves, the bottoms of those leaves had a lot more glitter than the tops. And this is the tops. So this is again glitter level 1 to 6, but on the tops, this is the older leaves, this is the younger leaves, and this was the exact opposite. So as the leaves got younger, they had more glitter on top. And this is a comparison of the two. So you can see how the bottoms of the leaves had a lot more like variety and difference between how much glitter they had in general. So I had some leaves that got up to a 6, and that's why I had to change my scale. But the tops had barely got above a 2.5. So it was pretty interesting to see that. So I took some averages. This is the top of the leaf, and this is the bottom. So you can sort of see the shininess over here that is the glitter. So the average glitter on the tops of the leaves was 0.41, and the bottoms was 1.48, and that's on a 1 to 6 scale again. So you can see how much more glitter there was on the bottoms. So my results for those 4 hypotheses I had was that it might be for insect protection. Nope. There was no correlation. It was just sort of they ate whatever they wanted. For sunscreen, nope. Again, the exact opposite occurred where I found there was the plants in the shade had more glitter than the ones out in the sun. So that was a little interesting. The third one was that it might present moisture loss. This is maybe leaves. Most of where they do water and all that moisture loss, they lose a lot of moisture on the bottoms. So with more glitter being on the bottoms of the leaves, that might help protect it. But again, as the leaves got younger, they had less glitter on the bottom. So I don't really know what's going on there. And my fourth one was that new leaves will have more glitter because they haven't been as exposed to the elements. And again, this was again wrong because the older leaves had even more glitter than the younger ones. So, my result's important because little is known about the function of the glitter on the autumn olives. So, while doing background research, I found literally nothing about what glitter is used for, what it was. It just said, to identify the plant, look for the shiny stuff. I was like, that's not helpful. They are extremely detrimental and invasive in the United States, so they're not helpful at all, really. I mean, they have berries, so they feed birds and stuff, but that spreads them more, and they take over and kill natives, so that's not really helpful. And fully understanding a plant is the key to be able to manage it, and in this way, we want to try to stop the spread of them. So understanding what this glitter is, if it's something to help it, then you can maybe combat that somehow. If I would do the study again, I would like to measure the soil conditions, again about the nitrogen and how much water was present, and sample in a greater variety to limit unwanted conditions, again like the sun and the wind. And I had a lot of thoughts about the moisture of the leaves. We spent a while changing and adding stuff, but I was very confused why the older leaves would have more glitter on the bottom, but the younger didn't. So, like, why do younger leaves have less glitter on the bottom when you think it would be really important for them to retain water because they're younger? But yeah, that was pretty interesting to see. So again, the scales or the glitter on all the model leaves is not used for bug retention or sunscreen. Younger leaves do not have more glitter than older leaves. In fact, the opposite occurred with older leaves having a bigger amount of glitter than on the bottoms and overall than the younger leaves. And the glitter may help regulate water loss. And these are my sources.
It's about more cores.
Hi everyone, my name's Lane Mason. I'm going into 10th grade. I've been homeschooled all my life, and this is my 4th year doing Young Scientists. So on Monday, Miss Bridget took us out on this hike, and she cored a tree, and we passed around the tree core, and I noticed that it was really, really wet, and I was wondering why it was like that. So I was wondering if different types of trees would retain different amounts of moisture in the tree core or just in the tree overall. And I also wondered if the pH would vary between the species of tree. So different trees, in my background research, different trees have saps with different pHs, but we didn't really know anything about the wood itself. And also in my background research, we realized that climate and elevation can also affect the pH of the sap. So my research question was, are different species of tree more wet than others? Does the wood vary in pH between species? And are wood moisture and pH affected by the soil pH? My hypothesis was that I thought the soil moisture would affect the wood moisture, and I thought soil pH would affect the wood's pH. So I spent the week in various forests around Clifton. This is a place called Adams Woods, which is up here on the map, is where that photo was taken, and these are the 3 different spots where I took cores. So, like I said, I spent the week coring and collecting soil samples. So we used a tree corer, which is this. It's got this orange bar, and this is the drill. And then I would use just a regular old trowel to collect the soil samples. So I would measure the diameter of the tree with a measuring tape and the help of Miss Cheyenne to get around some of the bigger trees. And we would use a GPS to figure out the elevation of the tree. So after taking the tree cores, we took them inside and measured the weight of the core. Then we would bake the cores, like you can see here, to remove the water and weigh them again. That way we could figure out how much of the weight of them was water. So we would calculate water weight as a percentage of wood weight. So the average percent moisture in the tree was 63%. So about 63% of the weight of the core itself would just be water. And if I pull these out, you all can pass this around. This is the tree core. So to take the pH of the wood, we smashed the cores with a hammer and let the flakes sit in water. And to do the soil, we basically did the same. We would just mix the soil with water. And then we were originally going to count the rings, and after we baked them, a method that we learned online was to sand them down a little bit, and hopefully then you would be able to read the rings. But unfortunately, it didn't really work that well. A lot of them are really hard to tell, so it just didn't work that well. This is a pH scale. If you do not know what a pH scale is, the more alkaline a substance is, that would be a base, and the more acidic a substance is, that would be an acid. So an example of a base would be bleach. This is like an 11 to 13. An example of an acid would be a dehydrated sour Skittle, which is a 2 on the scale. So this graph shows, because I said we were also measuring the diameter, this is the number of rings, roughly, because obviously we couldn't exactly get it. So you can see in the tulip poplars, there is a clear indicator that as the tree gets older, the diameter gets larger. In the pine, it's the same, but it's not quite as steep. So every year, a tulip poplar would grow about 3 centimeters, which is a lot, considering it's a tree. And a pine tree would only grow about 0.8 centimeters. So this shows water weight as a percent of wood weight. So you can see clearly that the tulip poplars had a significant amount of water, the most being about 124%, and the least being the pine, which had like only a tiny little bit of water. And actually, the third tulip poplar that we cored, there's this little hole down the middle, which is where you get the tree core out of. After we took the tree core out, I was turning it the other direction to pull it out, and water started gushing out of this hole. And I got soaked. It was great. So this is the pH of the wood and the different species of tree. So you can pretty clearly see that between species of tree, the pH doesn't really vary at all. That's tree pH, and this is soil pH. So once again, you can see there is no correlation between the tree pH and the soil pH. This is the water weight as percent of the wood, and this is the elevation. So this really surprised me. You can see that there is no correlation between elevation and how much water was in the tree. So my results showed that tulip poplars are the wettest of the tree species we studied, and pine are the driest. I found that pH doesn't vary at all between the different species of tree. Soil pH does not affect the pH of the wood, and elevation doesn't affect the amount of moisture. I couldn't find a lot of research about how much sap trees make and the pH of that, so my results might be new information. And I did find information that trees have different pH overall, so I was a little surprised that the tree species that we studied didn't have any of that shown. I was very surprised that the soil pH had no effect on tree pH, considering that's a really key factor in how much the tree grows, is the pH, the moisture, stuff like that. I also expected elevation to affect the amount of moisture, because you would expect that there would be more water and lower elevations. And also, I was very surprised how hard it was to turn the tree core in some trees, specifically hickories, which is how we may or may not have accidentally broken the tree core, but it's fine. And also, the pH strips we had were not very accurate, so if I could go back, I would use some more trusty equipment. And these are my references.
That's patches.
So, my name is Mila Hobb. I'm going into 9th grade and I'm a homeschooler. So, some of my hobbies include reading, playing volleyball, crocheting, hiking, and kayaking and camping. So, at the beginning of the week, I had no idea what to do. So, I just looked around and I saw that some flower patches had a really diverse range of all the insects on them. And so, I was really curious about the relationship between the insects and the plants. So, that was really broad. So I had to narrow it down to my project I was going to be studying this week. And I ended up on studying the number of butterflies visiting separate patches of milkweed. So just some background information that may have impacted some of the results is that on hot days, a butterfly might find a shady spot and fold its wings to keep itself cool. And different species of butterflies might only be active at different times of the day to keep themselves safe from various things like predators and heat. So I think that both of these things could have impacted how many butterflies we saw. So my research question was, does milkweed species inside the patch affect how often it is visited by different varieties of butterflies? So my hypotheses were that I thought the different species I measured, so the butterfly and the common milkweed, would have roughly the same results. And I also thought that the larger patches would attract more butterflies of different species. So to do my research, I had a lot of sites, wherever milkweed was really. So Turkey Gap, Milkweed Field, Upper Woodcock, Lower Woodcock, the road near the driveway you probably came up, and then Indian Grass Field. So those are all the white dots. And these habitat types were meadows and the edge of tree lines. So my methods to get my information was to locate patches, and then at that patch I would count the flowering milkweeds and record the conditions like the time, level of sun, and temperature. And then I would set a 10-minute timer for butterfly observation and count how many butterflies I saw and identify the butterflies. I couldn't exactly keep track of if the butterflies were the same butterflies that left and then came back, so I just counted butterfly visits. And then the tools I used were binoculars, field guides, and a notebook and pen. So some of the challenges that I sort of faced were the identification of very similar butterflies, especially at a distance, defining landing versus flying by, because sometimes there was like a piece of grass or something in front of it, and I couldn't tell if it had really touched down at the flower or just flew by. So I had to figure out the difference between those sometimes. And then butterflies appearing after the timer finished, which was really very annoying because I really just wanted to include them, but I couldn't. So. Some other variables include that I couldn't really keep track of were the distance to the milkweed, because I just wanted to sit in a shady patch to watch it. So I might have disturbed some of the butterflies, so that I feel like could skew the data a little bit. And then the data that I collected was the butterfly species counts and flowering milkweed counts. So on average, I saw about 5.65 butterflies per 10-minute watch period on each patch. So the overall distribution of the butterfly species, so this shows all the species that I saw, and as you can see, more than 50% of it was just small unidentified butterflies, like some of them were silver-spotted skippers and coral hair streaks. But some other important information off of this is that the great springled fritillary was the most frequent of the larger butterflies that visited, and then the least frequent large visitor was the monarch. So this is just a Venn diagram showing which butterflies landed in which type of milkweed. So the middle ones are obviously the ones that landed in both, and then there was actually a few that landed only in the common milkweed, which were the tiger swallowtails and a monarch. So this is just a graph of how many butterflies how many visits there were per type of milkweed. So on the bottom, it shows on the left that that's the common milkweed, and then the right is butterfly milkweed, and then the y-axis is the number of visits. So we can see that there were many more butterflies that visited the common milkweed patches compared to the butterfly milkweed. And then we also see that there's similar numbers of large butterflies on both milkweeds, with it being like a little bit higher on the butterfly milkweed, but not enough to conclusively say that there's definitely more larger butterflies on the butterfly milkweed. And then the majority of the butterflies were the unidentified small ones, but the distribution of the types of butterflies in different types of milkweed are very different. So about half of the ones in the butterfly milkweed were the small ones, and then about 2/3 of them were the unidentified small ones in the common milkweed. So this shows how many butterfly visits there were per plant. So here we can see that the common milkweed had more visits per plant, but the butterfly milkweed seemed to have more of a variation of how many visits there were per plant. So this is, in my opinion, the most interesting graph, because it shows how, as the total number of plants in the patch increases, the total number of butterfly visits also increases. So, yeah. And then my results were that the common milkweed seemed to have more total butterflies and a greater variety of butterflies, and also seemed to attract more butterflies per plant. Whereas the butterfly milkweed seemed to have higher proportions of larger butterflies, and then the larger patches tended to have more butterflies. So I think that the reason that there were more butterflies that visited the common milkweed could have been because maybe it was a preferred size, shape, or color to the butterflies. And I think that maybe a reason why the butterflies would go to larger patches would be possibly because there was just more nectar available at a closer range, so they didn't have to fly as far. And then just an interesting study that I found was there was a study of egg density on milkweed found, and this found that the more plants in a patch resulted in more eggs in a patch, which sort of goes similar to what I said, but is just a little different. And then these results can also help inform anyone looking for plants that attract butterflies. So just some things that are good takeaways if you're one of those people would be that both milkweed plants attract a large variety of butterflies, and larger plots attract more butterflies. So basically, plant more plants, and also both are good options. So just whatever is your favorite color. Then if I could continue the study, I would probably collect more data to compare the sun versus shade, because I didn't have very many shady spots that I could compare. I would probably try to regulate the temperature more between each section, and then I would also try to control the time of day a little bit more. So as I said earlier, I would also probably have a specific distance from myself and the patch of milkweed, because I feel like that I might have disturbed the butterflies at some points, and that might affect my data. And so I would also increase the observation time from 10 minutes to 20 minutes, because, as I said earlier also, a lot more would come right after the time. So I would also probably identify the smaller butterflies, because they made up more than 50% of it. And then last but not least, I would track other flowers around the milkweed that could impact my data. So in conclusion, I learned that my hypothesis was correct in that larger patches of milkweed did attract more butterflies and a larger variety of butterflies. And from my data, I can conclude that common milkweed attracts a larger variety of butterfly species, and common milkweed also attracts more total butterflies. And these are my sources.
Plant preferences.
Hi, I'm Alistair. I'm going in 10th grade at Wakefield. I like spiders and biology. So, as some of you may know, spotted lanternflies are slightly invasive, and they're from Southeastern Asia, and they're specifically damaging to crops and fruit trees along with native trees. And so when we went on our walk on Monday, I noticed that lanternfly larvae or nymphs were on more plants than others. So like some would be more on this one than on the lower one. And I was like, that's interesting. So my question became that. And then scientists already know that lanternflies like Atlantis, which is their host plant, also known as tree of heaven, and they also like black walnuts and wild grapevines. So my research question was, what kind of plants do lanternflies prefer? And my hypothesis was, if a plant has similar leaves to Atlantis, then more lanternflies will be present, as Atlantis leaves are soft and compound. And so my study site was up in Adamswood over that way, where I noticed that there weren't that many, as I only got 11 from one plant, and only 11, and that was it. And then the rest of the week I was over in Lower and Upper Woodcock over that way, and then in the Upper and Lower Pond behind us. And a lot more were present there. And I was mainly in forest edges, and fields, which were more prevalent. And then they don't have much of a presence in forests. So Clifton Institute, I was in the light blue arrows. I don't know if you can really see them, but I wasn't in that circle. But I didn't really go anywhere that much. I just kind of was in one area for a while. So yeah. So these are simple versus compound leaves. Compound leaves look like multiple leaves, but are really just one leaf with multiple little outgrowths. And then this is a redbud leaf, and it's a simple leaf, and that's also redbud. So my methods of studying lanternflies were walking along one side of the trail and looking at the leaf or plants and counting how many lanternflies I saw, if I saw any on plants. But I was also looking for specific trees and plants. So I was looking for compound trees and vines. I was looking for ashes and Virginia creepers. And for simple trees and vines, I was looking for redbuds and wild grapevines. So I would be walking along the path looking for those plants, and then if I saw any, I would note them down and then try to see if they had any lanternflies. It wasn't successful most of the time. I had an entire page full of redbuds and only about 17 lanternflies from 2 plants. But I just did that for most of the week and then continued. I made this colorful mess out of hot glue, a dream, and some pipe cleaners. And it was to see if lanternflies preferred rough surfaces that also had fluffier stems, so softerness, that's a word, compared to slicker, softer branches. And I set those out on Thursday in the morning, came back during lunch, and to the grand surprise of this all-new natural tree, there were 2. Only 2. On this one right here, so the rougher blue one, and the more natural-looking green one. And that was definitely worth about a day, and then waiting 3 hours. Yeah. So these were the averages of the plants and plant texture and number of lanternflies. So on rough, it was about less than 20, with most of them being less than 10. And then on smooth, I had 1 tree, 63 lanternflies, and it was probably the most vile thing I'd ever seen. But it was mostly around 15-ish, maybe 10, for most normal times. So type of leaf, so simple versus compound, same issue, 60 on the same tree, and then a bit more than 20, but mostly 5. And then number of lanternflies on specific plants. So ash had the least, well, almost the least, but it was tied with redbuds. But it typically had smaller amounts but more across its leaves than than the other ones. But grapevines had the most, as they were a mix of smooth leaves but a rough tree part. And then red buds were about the same as ash, just more. I mean, the simpler leaves and smoothness. And then Virginia creepers had the least, and they were only there because I didn't know what else to look at. So my results were the nymphs prefer soft, simple leaves over rough compound leaves. And, oh dear. No, that's fine. So based on my results, lanternfly, the nymphs prefer softer leaves since the texture is more similar to ailanthus. And, yeah. So then, my background research also stated that they like softer leaves, so black walnut leaves and grapevines. And so the reason you should care is if we know what plants are liked by them, it'll prevent the spread of the further spread into the country. And my results are interesting because I spent a bunch of time looking at plants and being interested. It was a fun project, but counting the lanternflies was quite interesting because I'd look at a plant, look at it again, and see, hey, look, 5 more lanternflies after previously having marked down 0. But if I were to do this project again, I probably wouldn't focus on 4 trees that I'm barely able to identify because I'm bad at plants. And I'd mainly just be like, oh, that vine has 15 lanternflies on it. And if I continue the study, I'd probably just look at a more broader sense about like what plants I see and if they have any lanternflies on them. Because most of the plants didn't. It was mainly just the taller trees and some shrubs that they fell on because they're really good at climbing. But in conclusion, nymphs seem to prefer smoother surfaces over rougher surfaces, and you can ask Bridget about the orange bucket.
I sure can.
Yeah. And they also seem to prefer simple leaves over compound leaves, even though their best friend, the tree of heaven, has compound leaves. And these are my references.
Evan, rising 12th grader, homeschool dual enrollment at Germanna Community College. This is my second year at the Clifton Institute for the science, young scientists research. My hobbies are slow hikes, sports, fishing, and wildlife photography. I love birds, and I want to understand how I can better attract them, how I can find them better. And so, by comparing what insects like to attracting the insects to attracting the birds, that's kind of my focus. So my question was, do birds prefer native species of plants or non-natives? And I thought most species of bugs like our native plants because they've evolved with them for hundreds of years. But non-natives are new, and I wouldn't expect them to really use them that much, but it's kind of surprising. So native plants are any kind of plant that has been here that is not taken over by overseas by any people. So they've been here, adapted around each other, created an ecosystem together. A non-native plant is something that was brought over from somewhere to help us, and like they brought something over to like prevent erosion that ended up like taking over forests. So that creates invasive species, which will take over an entire forest and become a monoculture, which is not good. But what the good ones are keystone plants, and they're keystone species because if you remove them, you can essentially lose your entire ecosystem. So bugs, there's specialists, and then there are generalists. So specialists have evolved around the species of plant. So like a monarch butterfly on milkweed, you need to plant milkweed to have monarchs because they lay their eggs on the milkweed, and the caterpillars grow up on that. They will only lay their eggs on milkweed. So butterfly weed and milkweed. And then there are generalists, which will use anything that they can find, opportunistic. So they'll go around and, like true bugs, which the lanternfly is a true bug, they will literally suck out of the plant, suck out of animals. They're generalists. They take opportunity of as much as they can. So just true bugs are any bugs that have mouth parts for extracting nutrients from animals and plants. So where did I do my research? On the first day, we went down to Turkey Gap and did some research along the forest edge line of the center and then kind of around the edges. The second day, we went up to Upper Woodcock, did the edges there. And then the last day, we went down the Indian grass field where we did some research around the forest edge lines over there. So habitat locations, Turkey Gap, Upper Woodcock, Lower Woodcock, Indian grass field, and then habitat type was edgeline. So we have meadow on one side and then forest on the other, kind of where it meets. I used shrubby. I tried to pick shrubby plants for my project, and it's kind of a shaded but exposed area, so there's some sun, but not too much sun. My methods was I would take a white sheet and I put it underneath the plant, and then I would get, if you look over there, there's a net where the wooden handle is, the really tall net next to the buckets. I'd use that handle and I'd whack the tree, and it would shake it. And my hope was that any bugs would get shaken off the tree and fall onto the mat. And then I'd count them as quickly as I could, because otherwise they would all run away and jump off. And I also had to be careful because there were other bugs that like to jump onto the mat, so I had to make sure I wasn't counting those guys. The challenge is there were a lot of variables. So some of this data might not be the most accurate, but time of year, hit forest, shrub size, varieties of shrubs and trees, so like how big they are, how many branches they have, how many leaves they have. The bugs can fly away. Some will have wings, and they will, as you whack it, they just fly away, like pollinators and stuff. So I didn't get many bees. Some of them, like the little caterpillars, are able to hold on pretty well, so they don't always fall off. Another one is miscount and misidentification. First part of the week, I was IDing a lot of them as bugs or beetles when they were actually true bugs. When it turned out they were beetles, but they were so small I couldn't tell. So the data collection I'd find 4 of one species, so I'd get like four autumn olives all along the forest edge line, and I'd whack 4 different plants, 4 different oaks, all sorts of stuff, and then count them on the bug tarp, and then categorize them by their order. The average bug shaken off the plant was around 7. It's like 6 point something, but you can't have 6 point something bugs. Most bugs found on the plant was true bugs at 68, and the least found was isopods. I found one little roly poly that fell off a branch, which was not supposed to be there, but so this is kind of the species of tree right here, and then how many of each individual, and then the species itself. So these are all the natives and then the non-natives, and it surprised me. The least amount was actually on a native, red bud. Now, do not think red buds are bad. They're actually amazing. It was just a time of year. And sadly, they had a lot of ticks on them, which is not a good reputation, but privet was not very useful, but autumn olive actually had a lot of bugs on it, which was surprising. A lot of ants. So some of my observations was the true bugs and bees and ants were 50% of both, at least. And then spotted lanternflies were about the same on both non-native and native species. And then plants, native plants hosted 2 times the amount of beetles than the non-natives. So that could just been because of my counting and IDing them wrong, but I think it'd be the same all around. So there wasn't much of a difference between native and non-native hosting the amount of bugs. Something that could have changed it was time of year. Only one of the species was flowering, and that was the sumac. So, but the problem with that was, again, the pollinators all have wings, so whenever you whack it, they all just fly away. You don't really catch them. So that definitely could have been an impact on that. And then I think that if I'd pick more flowering plants, more insects are using those at a time. So I think that maybe I would have gotten bigger numbers if I had to pick all flowering plants. My results was highest bug count. So on the pin oak and autumn olive, those had the most. Autumn olive had a lot of ants. Oak had more beetles. Sumac was third place for most beetles. Sumac had a high number of true bugs. I didn't catch any pollinators exactly because they fly away. Redbud likely due to time of year because they bloom. They're our earliest bloomer, so they're out there for like the early stages of the bees. So all those bees that come out early have something to actually pollinate, but because it's late in the year, I don't think they have as many bugs on them. Everything has moved to more opportunistic places. Issues, again, too many variables, time of year, expectations. I expected to see a difference in the total bugs between native and non-natives because, again, natives have adapted here for much longer. The bugs have had a more chance to understand what tastes good and what doesn't, but not really a difference. If I could do this project again, I'd, again, pick all flowering plants as possible. And if I could study this more, I would do it in like different seasons, like fall, summer, winter, all that. And then in conclusion, I learned that bugs seem to have a preference with the natives versus non-natives, and then I would need to use a different method for different variables. Have references.
Vireos and scarlet tanagers.
Hello, I'm Luke Theodore. I'm a rising ninth grader at Wisdom High School, which is in Fairfax, which is further east. I enjoy drawing, birdwatching, and running. So my motivation for doing this project is I like birds, and I was intrigued by the Red-Eyed Vireo song. I kind of liked it, and I wanted to see if there was like a pattern I could pick up on. Scarlet Tanagers kind of have a similar song pattern. They don't have similar songs, but they kind of all have the phrasing that's similar. Similar phrasing with pauses in between. So yeah, the birds I was studying, Scarlet Tanager and Red-Eyed Vireo, and they both have songs that are like individual warbled phrases followed by a pause, and then they keep going, and then they stop, and then wait a little bit, and then go again. So the phrases are making up the songs. So my research question was, how did the songs of Red-Eyed Vireos compare to the songs of the Scarlet Tanager? And my hypothesis was both species will have a sort of pattern I can pick up on. So a certain like a one or two series of phrases that they repeat over and over again. Okay, so my method was I visited many forested locations with breeding birds, and I used the Merlin app to take 5-minute recordings when we heard either of the species. I got the privilege of visiting most of the forest here. All the pink stars are places I took recordings. And to analyze the recordings, we'd come back inside. This was very tedious, but I would listen to all the recordings. And then for each species, I had a library that looked something like this. And each letter was assigned to a phrase, kind of thing. And then I had other data on it. And so as you can see down there, that first one is an A, B, C, D, E, F. And that's a Scarlet Tanager song. Mm-hmm. So this is my first piece of data, and this is the number of phrases in the song of the birds. So as you can see at the bottom there, Red-Eyed Vireos are a lot more varied in the length of their song, having a range of 14. That means the length of the longest song minus the length of the shortest song is 14, while Scarlet Tanager is only 9. Okay, so the rest of my project is going to be using a lot of transition matrices, and this is how you read them. So this is Scarlet Tanager number 1, okay? So you start on the left, and you pick a row, and then so if you pick C, and then go across, it says 92%, and it's very dark red, and that's in column D. That means when the bird sings C, there's a 92% chance that this next one is going to be D. And also the diagonal line that you notice, that shows a repeated pattern, because that means most of the time the bird starts with A and then goes to B and then goes to C and then goes to D and then so on.
Oh.
Yes. So here's another scarlet tanager, and as you can see, there is a noticeable diagonal line. It's not as clean as the other one, but this is also a very variable bird in terms of the number of songs it has. Okay, here's a red-eyed vireo. This one does not have a diagonal line present, meaning it's more or less randomly picking its songs as compared to the tanager. This one, similar story for this red-eyed vireo. It seems to prefer its NG and D songs, or phrases, in its songs, but it doesn't have any specific order it does it in. So my results were red-eyed vireo songs are much more variable than scarlet tanager songs, and they have a variable length. They don't have a specific pattern, whereas each individual scarlet tanager will have have its own kind of unique song that it will sing a lot. So my hypothesis that birds repeat the same song pattern over and over was true for the tanagers, but not true for the vireos at all. But my results do agree with some of the background research I did that showed that red-eyed vireos are very complex songs, and they're different between individuals. But that's all I got for background research. There wasn't very much on that topic. Red-eyed vireos could either be very sloppy learners, or they could be great improvisers. I don't know. That's up to you. Both of these species are songbirds, meaning that they learn their song generally. So my data is suggesting that the different songbirds have different methods of learning songs, because that's a very different in terms of how they're singing. And my project could have been improved by better recording equipment. I was just using a cell phone, and more time to catalog the sounds, because that took a while and that was tedious. I also could have, while I'm at it, include more species of songbird, because maybe there's even more ways, or maybe some of them have similar ways. No. So conclusion: red-eyed vireo songs are more variable than scarlet tanager songs. That pretty much sums up everything. I think that's cool.